my name is Viren Jain. I'm coming here from Google today. So I'm going to talk about Connectomics. I'm going to focus most of the brief talk, about the computational challenges in general. And then I'll talk a little bit about a recent effort started about 8 months ago to try and address some of these challenges at Google using some of their infrastructure and resources. So we just heard a talk about the mesoscale connectome. So that's about projections and pathways in the brain. The kind of data and objective that I'm going to talk about today is more about synaptic level circuitry. So really trying to recover, at the level of microcircuits, what is the actual wiring diagram of those individual circuits at synaptic resolution. And in general, this involves, taking some tissue of brain, slicing it up, imaging it with an electron microscope, segmenting and reconstructing the data to produce finally a circuit diagram, where again, each node here is an individual neuron, and some entry in that matrix is a particular synaptic connection or weight between two neurons. And just as an example of sort of the experimental paradigm, so here's a recent study from Kevin Brigman, Winfried Denk at the MPI, where they first imaged a piece of retina in a mouse using 2-photon calcium imaging to measure direction selectivity of specific neurons. They took that specific tissue, which was imaged functionally, imaged it using an electron microscope, in particular a technique called serial block face SEM imaging, and then were able to identify those cells they functionally characterized in the structural tissue and begin to map out the underlying anatomical circuitry of those functionally characterized cells. So here you're seeing a tracing, in this case done by humans, of one specific cell within that piece of tissue. And as we begin to zoom out, you can sort of see how intricate the neuropil really is here. So this is mostly the inner plexiform layer of the tissue, and that was the cell body, and now we're seeing the neurites sort of extend out. And again, this is just one single neuron in this field of view, and then we'll see others come into the field of view later on. But in any case, you get the idea that there's an immense amount of complexity and data here, and ultimately, analyzing this kind of data is going to require lots of help from computers. So the basic thing we want to know from this type of data, from the anatomical portion of it anyway, is which neurons are connected to each other, right? So in order to answer that question, there's really two computational tasks that have to be solved. The first is a segmentation problem, so taking the raw data, like on the left, and tracing out the individual neurons, like has been done on the right. And the tracing here that's being shown was done by a human, at a cost of 50 to 60 hours per neuron. And this is not even the entire neuron, just whatever's contained within the field of view that was imaged. So clearly that's not a process that's going to scale based on human effort alone. So this is one side of the problem, tracing the neurites. But to actually find a synapse, you need both the structure of the neuron as well as local evidence of a specific connection between two neurons. So this is sort of a canonical synapse. Here you see the vesicles and a postsynaptic density. And so this is sort of the object localization part of the problem, where you have to go and find these synapses to establish a real positive connection between two neurons. And the challenge part of this is really the following, that conventional image processing or even modern techniques based on machine learning and so on don't solve the problem. They make too many mistakes. And so what do we mean by a mistake? So here's a connectivity matrix, right? So the columns and rows here are different neurons, and then non-negative entries here represent connections between two different neurons. And so you can imagine a false positive, right? So you put in a connection where it doesn't really exist. You can imagine a false negative. You put in a connection where, you miss a connection where you shouldn't have. If our error rates were sort of limited to isolated cases of these false positives, false negatives, we might be okay, right? I mean, even in genome sequencing, they still have a small error rate, and certainly earlier techniques used to have a much larger error rate. So if it was just a few percentage of the connectivity matrix that we were missing, we might be okay. But the situation is such that modern techniques make so many mistakes that you get something that's completely useless, right? So we really need significant advances in the image processing techniques in order to generate something that's useful for this problem. So in summary, that's the first challenge, right? So how do we increase the accuracy of image analysis as related to this problem? So the second problem is really related to scale. So in the 70s, people sliced up, Sidney Brenner's colleagues at the MRC sliced up a C. elegans worm imaged it using electron microscopy, specifically transmission EM, and then they hand traced the images over a process of cumulatively around 10 years, and that was for about 300 neurons. More recently, this sort of study has been revived using much more help from computers. So for example, a group from MIT and the Max Planck recently did a study of the retina. That was about 1,000 neurons that was reconstructed. In progress are even more ambitious studies related to reconstructing a cortical column or a whole Drosophila brain, which would be on the order of 100,000 neurons. And some really ambitious people, like Winfred and his colleagues at the MPI, are trying to design microscopes to do even a whole mouse brain, right? And the data sizes here scale quite a bit, right? Because this is three-dimensional imaging, and each voxel is in the nanometer range. So the current data sets that are being planned are in the order of a petabyte, and for something like for even a significant fraction of a whole mouse brain, it's many petabytes. So where's the scale coming from? So there's actually some quite interesting advances on the microscopy side. So what I'm showing you is an image of what's called a multi-beam scanning electron microscope. So a typical microscope for this application has a single beam and a single detector, and that's great unless you want to image even more tissue. So people are now pursuing methods. So this is actually an image from a microscope that Zeiss is working on, where there's simply 61 beams instead of one beam, right? And you can imagine that if you do that properly, then you get quite a factor of scale up in the amount of data you can collect. And so the projections at this point for this kind of imaging and other systems is on the order of 100 terabytes per day, which might not actually sound so bad, except that the image analysis involved here is quite intense, right? So the image is completely dense. We can't just ignore large parts of it. And in order to actually segment and reconstruct it, we end up doing quite a lot of computation per pixel. So that number is actually a little bit concerning. So in summary, challenge number 2 is how do we store and process streams of data that are this large? And the final challenge, which I'll talk about, is something which actually is not that highly discussed these days, but I think is going to be a very significant problem, which is the actual analysis of any sort of data like this. So currently, most of these studies are sort of n equals 1, right? You take a single Drosophila brain and you spend a whole lot of money and effort to reconstruct it, or a single cortical column, and you do the same, and you get a single wiring diagram out of that, and that's great. That's sort of the place where you start, obviously. But if you look at, for example, genomics, a lot of the power has come out of comparative approaches, right, where you sequence many different specimens, many different species, and then you compare them. You develop databases and you find ways to find differences, find similarities. And it's quite possible that connectomics will be similar in that you'll want to measure connectomes under various different conditions, under various different circuits and different species, and find ways to measure differences and similarities. And so this third challenge that I'm mentioning is really how do you organize and query and analyze large amounts of wiring diagram data? And that's partly an intellectual problem. What's the right way to pose that problem and mathematically formulate it? But it's quite likely to also be quite a significant computational and data science challenge. And so that's something which I hope more people will be inspired to start thinking about, because at present it's sort of unknown what to do here. Certainly there's a lot of generic techniques which you can imagine borrowing from network science and other areas, but as related to neural circuits and computation, it's not clear those techniques will be most appropriate. Okay, so the second part of things which I'm going to talk about here is there's various issues in storage and computation and algorithms, which are mentioned here and which I just talked about. And so one question is, well, can industry help? Right, so the internet industry has basically provoked a lot of development of data analysis tools and infrastructure, and so it seems natural to ask whether we can sort of use some of those tools to solve some of these problems. And given my affiliation, I guess you can guess the answer, but I guess I'll make the case now. So in terms of storage, petabytes are not a problem, but that's sort of the first order issue. The more serious issue is, you have to organize your storage in a way that you can compute with it in the way that you want, so that it's scalable. And of course, there's various use cases, right? So batch computation, visualization, serving, and distribution. So for example, at Google, we've been developing a storage system designed specifically for volumetric image data on the petabyte scale. You basically chunk up your data and then organize it using various kinds of space-filling methods. But the real core concept here is to turn everything into a key-value storage problem. And for those of you in the data science world, I'm sure that's not really a surprising solution. Obviously, some of the subtleties matter. But basically, any sort of large-scale data computation platform at this point works based on these key-value storage schemes. And so the first thing we did was basically design a way to store our data in a way that's compatible with that. And then once you do that, you basically get a lot of things for free. So for example, we can batch process our data using Google scale infrastructure, more or less just by virtue of the way that we've stored the data. The computational frameworks sort of come along for free once you've done things that way. So in terms of computation, of course, it's not yet clear what exact algorithms are going to be required to reconstruct the data. That's sort of still an area of research. But there are some things that are known. I mean, it's probably going to involve various kinds of feature detection and deep learning and graph-based algorithms. And so in order to do any of this, it's really important that your computation is co-located with storage. And again, that's another thing which I think modern data analysis platforms, whether it's at Google or elsewhere, have to some extent solved, but is really important in order for any of these pipelines to really have a stand a chance of scaling. And specifically in relation to computation, there is some precedent for organizations like Google playing a fairly large contributing role to these efforts. So there was a project at Google called Exacycle, which basically provided almost zero cost, very large scale CPU resources to the scientific community for sort of embarrassingly parallel problems. And this project actually contributed, I think at the end of the day, over 1 billion core hours of CPU time to various protein analysis problems, et cetera. And these were resources that were basically spare time at Google because they were very low priority cycles. So there is some precedent for companies sort of making a big contribution to certain areas of scientific research in this way. And finally, the last thing I'll talk about is algorithms. And in some ways, this is probably the most significant because algorithms it's very difficult to find good new ones. And it's the one thing you can't necessarily just count on Moore's law solving for you. So the specific thing I'll talk about is in deep networks because they actually have a big role in connectomic reconstruction. And of course, the larger story here is that they've significantly advanced the state of the art in areas like speech recognition, image interpretation, and so on. And because of that, well, you can debate the causality, but in any case, corporations like Google and so on are now investing quite a bit in advancing the state of the art of these methods. And the claim I'll make here is that actually the connectomics community can make immediate and almost direct benefit from all of this work. And so deep learning actually has a little bit of a history in connectomics. So myself and collaborators have introduced a number of methods for segmenting data, connectomic data based on deep learning methods. Here's an example of a network architecture that we used a couple of years ago to reconstruct about 1,000 cells in the retina. But of course, the broader story here is that a few years ago, Jeff Hinton and his colleagues really brought deep learning techniques like convolutional networks back into the forefront by achieving extremely good performance on problems of more general interest, right, like image recognition from natural images. And my example of a network architecture isn't showing up. But in any case, they came up with a network architecture that was in some ways actually pretty similar to what was invented in the 80s, but now augmented by massive amounts of computation via GPUs. At Google, there's been an effort to parallelize training of these types of architectures using both CPUs and GPUs. And if you want details, there's publicly available papers about this sort of work. And this has inspired not only systems level work to advance the capability of training these architectures, but also novel architectures themselves. So recently there was a paper from Google as well as other collaborators on a new type of architecture for doing image recognition, which significantly improved the state of the art on natural image recognition via the ImageNet challenge. And here's a picture of the architecture. It's actually pretty complicated. You can read about it in the paper. And the point I'm trying to make here is that actually we've sort of taken these architectures and applied it to our data with very little modification and seen significant improvements. And so this is important, right? Because this means that all the work that's being done to advance performance on these other problems is actually highly relevant to a more niche problem that maybe wouldn't see the same attention. So just a few words about the general effort. So about 8 months ago, there was an effort started at Google to try and solve the computational problems associated with connectomic reconstruction. We're working with the Allen Institute, HHMI, Max Planck, and others. And obviously, we're trying to take advantage of all the infrastructure that's being developed for data analysis and machine learning, along with some new ideas specifically for this problem. For now, I mean, we're working with very specific collaborators. In the long term, we'd like to find a way to expose some of these tools via APIs to other research for broader usage. And finally, I'll just mention this is a collaborative effort by multiple people at Google. Peter Lee is actually in the room, as well as Michael Januszowski. Thanks.